We'll now proceed to our next finalist team, which is EquinOx from Johns Hopkins University.
Hi, we're Team EquinOx, and we aim to prevent delays in detecting hypoxemia among patients with darker skin tones. In every hospital, there are devices called pulse oximeters that measure blood oxygen saturation levels and are critical indicators of a patient's respiratory health. The need for these devices has rapidly increased during the COVID-19 pandemic. Studies have shown that even hospital-grade pulse oximeters are biased and overestimate oxygen levels for patients with darker skin tones. Consequently, medical providers need a more accurate method to measure blood oxygen saturation levels for individuals with darker skin tones in order to prevent delays in the diagnosis of low blood oxygen, also known as hypoxemia. Let's take a look at how pulse oximeter bias can affect patient care. If a dark skin tone patient develops hypoxemia, but their pulse oximeter reading is overestimated, only after a delay of up to 16 hours will the pulse oximeter measure a value low enough to trigger clinical intervention. This delay in care greatly increases the risk of lung and heart-related diseases.
So not only are pulse oximeters inaccurate for darker skin tone patients, this inaccuracy is especially impactful in low and middle income countries that have a high proportion of dark skin tone patients. Additionally, the most affected countries don't currently have the resources to deploy expensive pulse oximeters at all points of care. Taken together, these discrepancies have created a perfect storm that stands to affect the diagnosis of hypoxemia for billions of people worldwide. EquinOx has been designed from the ground up to be a competitive product in the emerging markets we hope to operate in. At $25, our current design can be made for well under the WHO recommended $40 for market penetration in low resource settings. Additionally, EquinOx is durable and easily sanitized, a must-have for low resource settings. EquinOx is as fast as current pulse oximeters on the market, and of course, our proprietary sensor and machine learning model account for differences in skin pigmentation. This means that all patients will receive highly accurate, personalized measurements that eliminate the bias found in other pulse oximeters. Here's a demo of EquinOx in action. EquinOx uses optical sensors to account for skin tone-based inaccuracies in the oximeter's calibration phase. Our simulations show that our solution produces a reading that is 2.8 times more accurate for detecting hypoxemia in dark skin tone patients. Having completed the prototyping phase, EquinOx is obtaining IRB approval for a clinical study at the Johns Hopkins Hospital to validate and improve our current algorithm. One limitation is the lack of non-essential features, given the low price point we're targeting. Additionally, like all pulse oximeters, EquinOx has trouble in low perfusion and low temperature states. However, given the current lack of innovation in the space, we believe that EquinOx is uniquely poised to succeed. Although one competitor claims to account for skin tone differences, this claim hasn't been clinically validated. Looking to the future, after validating the algorithm in our study, we will seek FDA approval, which will allow us to rapidly seek approval in new markets and deliver our solution globally. With our innovations, EquinOx will bring effective, economical pulse oximeters to everyone, empowering clinicians to provide equitable care worldwide. Thank you for your time.
Excellent. We will now open up the floor to questions for Team EquinOx. Elizabeth, I see your hand is raised.
Yeah, hey, I can kick it off again. Thank you. Really important technology and, really timely, I think. This has been a big discussion in the medical device arena for, quite a while. I'd love to know what you can share about the unique aspect that allows you to read through a darker skin tone. So, what are the aspects around the sensor and the machine learning algorithm that you're able to share that lead it to being 2.8% or 2.8 times more accurate? And then also just knowing a little bit, where's that 2.8 number coming from at this point? Thank you.
Yeah, I could start to answer some of those. So for the 2.8 number in particular, we ran Monte Carlo simulations to understand how our algorithm would fare when analyzing when doing pulse oximetry in like a representative population. So we generated over 1,000 models of fingers that follow population distributions in terms of melanin content and other physiological parameters. And then we applied our algorithm to that and saw that first of all, the racial bias was reproduced even in the simulation. and our algorithm could take care of that. And in terms of how EquinOx is actually working about it, we're using the optical properties of the dermis. So we have some additional sensors that are shining lights into the dermis and understanding how they respond, like how the melanocytes concentration varies, and other properties, how the perfusion varies. And then based on that, we apply corrections to our initial results that bring us closer to the true arterial blood gas concentration.
Great, thank you so much.
A question from Dr. Centuria in the chat. What is the gold standard you are using for validation? And how does the personal validation work in the field?
That's a good question. So it's currently a gold standard for measuring blood oxygen saturation. It's called the arterial blood gas analysis test. And it's mostly done in the ICU setting. So as part of this, we are planning an IRB study. We've already submitted IRB to Johns Hopkins Hospital, in which we will go to the ICU, place our device on ICU patients, and while they get APGs drawn, we can compare our device outputs compared to this gold standard measurements. And that way we can have a very objective measure to make sure that our simulation predictions of accuracy are in fact true.
Thank you. And I see a question from Dr. Wilson in the chat. Do you have an estimated cost of manufacture?
Yeah, so currently the additional sensors we plan on adding shouldn't add too much cost compared to current conventional pulse oximeters. So for manufacturing, we believe that the whole device could be manufactured for under $25. Including, you know, additional in the end, what the final retail price would be, that is still not really been considered yet, but the manufacturing could be done for under 25.
Thank you for sharing that. I see a follow-up question from Dr. Machen in the chat. Are you still able to get a valid reading with movement?
That's a great concern. So we had designed a custom silicone molded covering for it. So, and we found in our testing that even when you like move your hands around and whatnot, we should be able to get signals from the system. And then redesigned this silicone mold based on population, like kind of like considerations for different population sizes of fingers. So it does provide a tight fit that can ensure consistent measurements across movement.
And just to add on to that, we also have traditionally pulse oximetry is done with 2 LEDs to determine the concentration of or the oxygen saturation in the blood. But our device is also going to include some extra lights as well to help determine like a green LED, which is normally used for calculating motion and removing artifacts from that. So we should be able to use, you know, conventional motion elimination algorithms given our the current parts of the device. So hopefully that would result in more accurate measurements as well.
Dr. Green, I see that you're unmuted. Did you have a question for the team?
Yeah, just a couple. Can it be used in a neonatal ward, and how temperature sensitive is it? Oximeters are notoriously difficult in cold environments.
Our device is also, it packages a temperature sensor as well. So like most traditional pulse oximeters, it should be able to modify the calibration based on the current temperature of the patient. But as you mentioned, that pulse oximetry in low perfusion and low temperature states is a very difficult task for even high-end pulse oximeters, just because of the nature of how the blood is sitting in the capillaries in the hand. So we don't expect too many improvements at that fundamental level. Rather, I think EquinOx is more targeting improving the race-based discrepancy, or decreasing the discrepancy across different skin tone types.
Good luck.
Thank you so much, team. I think we'll need to keep moving to our final semifinalist team, but I do ask you to please answer the questions in the chat at your convenience.